Next, we will see how the genetic code was established. So, given the existence of 20 amino acids, but only 4 bases, groups of several nucleotides must somehow specify a given amino acid. So, 20 different types of amino acids are present in the protein, but only 4 different types of bases are present in RNA or DNA. So, how these 4 bases could code for 20 different amino acids? So, initially it was proposed that a group of 2 bases can code for one amino acid, but in this way, it can only specify 16 amino acids. So, from 1954, most attention was given to how triplets or group of 3 nucleotides might work, even though they obviously would provide more permutations, that is 4 into 4 into 4, that is 64, than needed. Only 20 are needed, but 64 possible combinations can be made. Each amino acid was specified by only a single triplet. It was assumed that successive groups of nucleotides along a DNA chain code for successive amino acids along a given polypeptide chain. So, a specific sequence on the DNA specifies a specific sequence of amino acids in the protein. An elegant mutational analysis on bacterial proteins carried out in the early 1960s by Charles Janowski and Sidney Brenner showed that collinearity does exist. In fact, collinearity means the linearity between the base sequence in DNA and amino acid sequence in proteins. So these sequences this show some collinearity. Equally important were the genetic analysis by Brenner and Crick, which in 1961 first established that groups of 3 nucleotides are used to specify individual amino acids. But which specific groups of 3 bases or codons determine which specific amino acids could only be learned by biochemical analysis. The major breakthrough came when Marshall Nirenberg and Henrik Matthey, then working together, observed in 1961 that the addition of synthetic polynucleotide poly-U, that is U, if this synthetic mRNA is added to a cell-free system which is capable of making proteins, that leads to the synthesis of polypeptide chains containing only the amino acid phenylalanine. So obviously it was clear that if only we are using U, so U will code for phenylalanine. So U code was specific for phenylalanine. Use of increasingly more complex polynucleotides as synthetic messenger RNAs rapidly led to the identification of more and more codons for the specific amino acids. Particularly important in completing the code was the use of polynucleotides like AGU put together by organic chemist Har Gobind Khurana. So, with the efforts of Har Gobind Khurana, it was possible to go further for the establishment of the genetic code. And one information I give to you, Harbind Khurana was initially associated with Government College University, Lahore. In this way, more specific sets of codons were tested. Completion of the code in 1966 revealed that 61 out of the 64 possible permuted groups corresponded to amino acids, with most amino acids being encoded by more than one nucleotide triplet. So here in this picture you can see this is the genetic code. So this is letter 1, first letter of the genetic code, this is second letter of the genetic code, and this one is the third letter. So first U, second U, and third then U. So U codes for phenylalanine. Similarly, UUA codes for leucine, and CUU codes for leucine. Similarly, AUC, AUA codes for isoleucine, and so on. So here in this table you can see these amino acids, almost all the amino acids, they are coded by more than one type of genetic code.